Welcome to Circuit Buddy. Today we'll be discussing about steroidogenesis. This is the second lecture on steroidogenesis. In this lecture, we'll be talking about the cortisol synthesis pathway along with the sex steroid synthesis pathway. Okay, the sex steroids will be of two types: the male sex steroids and the female sex steroids. The male sex steroids are also called the androgens, which are 19 carbon compounds, whereas the female sex steroids, which are also called the estrogens, are 18 carbon compounds. Okay, so now we'll be talking about the synthesis pathway in a very detailed way. First of all, let us see how the mother molecule looks like, as I have shown you in the first lecture. Let's see. This is cholesterol and this is the mother compound from which we can derive any type of steroid hormone. Okay, so you can see that this is a 27 carbon compound and it has a steroid nucleus, and this nucleus is also called the cyclopentanoparhydrophenanthrin ring. Okay, now you can see that this is the pi bond that was in between 5 and 6 carbon, which is in the B ring, and when we were synthesizing progesterone, this pi bond was shifted to the A ring. Okay, and there was a keto group introduced instead of this hydroxyl group. So we'll be talking about them, and this is the trick which we used in the first lecture. This is please doctor see all the patients. Okay, so P means first progesterone, and then we have the DC, that means the deoxycorticosterone. Then we have this C means the corticosterone, and then L means aldosterone. But in this pathway we will be talking about the cortisol synthesis pathway. So all the things will be like this, but we'll use this trick up to this portion. Okay, and this portion will be included in this lecture. Okay, so first of all, cholesterol, the mother language, is converted to the pregnenolone. Okay, so you can see that if you want to convert cholesterol into pregnenolone, you have to introduce this enzyme, and this enzyme is cytochrome P450 SCC, or you can also call it Desmoulins system. Okay, so SCC means basically the side chain cleavage enzyme, and what it does is to cleave this 6 carbon chain. Okay, and it is actually making it something like this. It is introducing a keto group in the 20th position. Okay, so you can basically see that the molecule is not a cholesterol anymore, it is pregnenolone. Okay, so pregnenolone. Now this enzyme is present in the mitochondria, as I've said you earlier. Now this pregnenolone is a 21 carbon compound. Okay, so let me write it here. It's 21 carbon compound. This pregnenolone was converted to the progesterone, okay, by the action of the enzyme 3-beta-HSD, but here you can see that the pregnenolone is first converted to the 17-alpha-hydroxypregnenolone, okay, by the action of the enzyme 17-alpha-hydroxylase. Okay, so what does it mean? 17-alpha-hydroxylase is means enzyme, and hydroxylase actually means it actually introduces a hydroxyl group to the 17th carbon of the molecule. So this is the hydroxyl group added to the 17th position of the molecule and producing the 17-alpha-hydroxy pregnenolone. Okay. Now this 17-alpha-hydroxypregnenolone is converted to the 17-alpha-hydroxyprogesterone. So first there is a little change in the sequence of reactions. So first of all, the pregnenolone is converted to this, and then 17-alpha-hydroxypregnenolone is converted to 17-alpha-hydroxyprogesterone. So you see very well, there will be the same enzyme, that is 3-beta-HSD, which will convert the 17-alpha-hydroxypregnenolone molecule into a 17-alpha-hydroxyprogesterone. And for this you have to introduce a keto group here, and this pi bond will be shifted to the A ring in between 4 and 5th carbon. Okay, now it is basically 17-alpha-hydroxyprogesterone, which is the first P. Okay, so P means progesterone. So this is in this session, not this. You don't have to take it into account. Okay, just keep it in your mind that there is a hydroxyl group in the 17th position. Okay, 17 alpha hydroxy progesterone, and then you get to the DC. That means you have to convert this 17 alpha hydroxy progesterone to the 11 deoxycortisol, and the enzyme is obviously the 21, okay, hydroxylase. Okay, so deoxycortisol, 11 deoxycortisol, and the enzyme is actually introducing the hydroxyl group to the 21st carbon of this molecule, making it the 11 deoxycortisol. So just write it here, 11 deoxycortisol. So by the name of this molecule you can easily understand that it is called 11 deoxy because an oxygen is absent in the 11th position of this molecule, and for this we have to introduce the oxygen, that is introducing a hydroxyl group here. Okay, so it is the same molecule being formed DC. Okay, DC, but 17 alpha hydroxy progesterone has been converted to the 11 deoxycortisol, not the 11 or 17 alpha hydroxy corticosterone or something like that. That will be incorrect. Okay, so you have to remember this, that from 17 alpha hydroxy progesterone we get the 11 deoxycortisol. Okay, so this is cortisol, which is also a 21 carbon compound. Now from 11 deoxycortisol, the next enzyme will be this 11 beta hydroxylase. Okay, so let's write it here, 11 beta, okay, sorry, 11 beta hydroxylase, which is forming the cortisol molecule. So what it does is actually introducing this hydroxyl group in the 11th position and making it the cortisol, not 11 deoxycortisol but cortisol. Okay, and this is the last compound in this series of reactions, and this is called the glucocorticoids. Okay, this is called a glucocorticoid, and we have seen in our previous lecture that aldosterone along with the corticosterone were called the mineralocorticoids. Okay, so first of all, this is the 21 carbon compound, and that is the end of this series of reactions. Now we'll be talking about the 6 steroids, how they are produced and how they can be converted from one to another. Okay, so for this let me erase this board and make it easy for you. Now here in this case the trick will not work, and but there is a very simple way to remember this. First of all, I call this a 17 column. Okay, so why is it called the 17 column? No one calls this, but I call this the 17 column for remembering purposes, so that I can remember it clearly and efficiently. So you can see that the cholesterol molecule has been converted to pregnenolone, and this is the structure of pregnenolone, but this is actually the 17 alpha hydroxypregnenolone, so just remove it first. Okay, now this hydroxyl group is also absent case of the pregnenolone. Now the pregnenolone molecule will be converted to the 17 alpha hydroxypregnenolone as before. Okay, so no change in that. The enzymes are also the same, you can see. Now after this, 17 alpha hydroxypregnenolone undergoes a 1720 cleavage by the enzyme 1720 lyase. Okay, and for this it is converted to the dehydroepiandrosterone. Okay, so it is now uh, androgen and androgen. So you can see that it has been clipped here, forming a keto group here. So it is a keto group here. Now it is called dehydroepiandrosterone. Okay, so it is not replicable anymore. Now it has become a dehydroepiandrosterone. Okay, so dehydroepiandrosterone, and the enzyme here working is 1720 lyase. So basically it is telling that it is cleaving the molecule in between the 17th carbon and the 20th carbon, making the dehydroepiandrosterone. And then the dehydroepiandrosterone is converted to the androstenediol. Okay, so it is converted into androstenediol. Okay, so here the enzyme is the 17 beta HSD, 17 beta HSD, which actually introduces a hydroxyl group here, okay, in the 17th position. So there is this hydroxyl group present, and now it is the androstenediol, androstenediol. Okay, now I used the 3 beta HSD to be the androstenedione, from which we can also derive the testosterone, but in this case we will be talking about the androstenediol, and after that we will be talking about androstenedione, which will be converted to testosterone and eventually to the estriol or estradiol. Okay, now dehydroepiandrosterone is converted to the androstenediol, which is converted to the testosterone. Okay, testosterone by the action of the enzyme 3 beta HSD. So you can see that if I introduce the 3 beta HSD, it would be like this. So the keto group is there, and this pi bond is shifted to the A ring in between 4 and 5. Now you can say that this is the structure of the testosterone. And now if I told you that if you just introduce the 3 beta HSD first, what will be the consequence? Okay, so it was like this, and if you just introduce this 3 beta HSD, again you will get this structure and this keto group, and this oxygen will be there. Okay, so this is not a diol, this is dione. Okay, this is dione. You can see this is the androstenedione, androstenedione. So now you can see that these compounds are all 19 carbon compounds, and this androstenedione can be converted to testosterone by just introducing a hydroxyl group to the 17th position, and it can be done by a very simple enzyme, that is 17 beta HSD, and you know this very well, and it is a very simple thing to remember, because we have been dealing with this for the last few minutes. Okay, so androstenedione is converted to the testosterone just by introducing 17 beta HSD. The hydroxyl group is introduced in the 17th position. Okay, now you can see that this testosterone is the male sex steroid hormone. Okay, so this is 19 carbon compound, and after that this testosterone is converted to the dehydrotestosterone, Okay, so actually it is dihydro, so not D, dihydrotestosterone, testosterone, by the action of the enzyme 5-alpha reductase. So let me write it like this: 5-alpha reductase. Okay, and this is actually more potent than testosterone, and its efficiency is more than testosterone. Okay, now this testosterone can be converted to the estrogens, okay, by an enzyme which is called aromatase, which actually introduces an aromatic ring in the molecule of testosterone. So basically this is now testosterone, and now you are introducing an aromatic ring here. So this is the aromatic ring, you can see, okay, and all the things like before, but the 19 carbon is not present here. These are the other rings. Okay. So you can see that there are two hydroxyl groups present in this case, and now it will be called the estradiol, which can be converted to the estriols in the liver, and from androstenedione you can also derive the estrogens. Okay, so this is the overall view of the steroid hormone synthesis pathway, and this is the last video on steroidogenesis, because I have talked about all the basic pathways that are there, but you can also get some other reactions here. For example, from pregnenolone you can get the progesterone, as I have told you earlier, and then you can see that if you introduce 17-alpha hydroxylase enzyme here, it will be 17-alpha hydroxy progesterone in this position, and then the 17-alpha hydroxy progesterone can be converted to androstenedione, and you know the basic pathway from here. Okay, so that's very simple, and I hope that you have enjoyed the video. Thank you very much for your support and for your kindness and your patience that you watched my videos, and please share and subscribe to my channel, and please comment whatever you like the most about these videos, and I'll be grateful to you for whatever you have done to me. You have watched the video for your benefit, and indirectly it has made me happy, and thank you for this. Have a great day.